Hello dear students, today we are going to learn about pulmonary surfactant and test for fetal lung maturity. Lung development begins in the embryonic stage between week 4 to week 7, and at this time the lung bud emerges from the foregut and differentiate into trachea and primary bronchi. This is followed by pseudoglandular stage, which ranges from week 7 to week 17, during which extensive branching forms and conductive airways form. However, at this time there is no respiratory bronchioles form or no alveoli form, so gas exchange is not possible at this stage. Next is the canalicular stage, which ranges from week 17 to week 26. And it is the critical transition stage towards the viability of the fetus, and during this period respiratory bronchioles and primitive alveolar ducts begins to form and vascularization increases significantly, as we can see in this diagram. And around this time, around 24 weeks, the type 2 pneumocytes starts producing surfactant. Although the quantity is initially insufficient for the effective function, but type 2 pneumocytes starts to form this surfactant. Next is the secular stage, which ranges between week 26 to week 36. At this stage, terminal sacs develop and gas exchange becomes possible. At this time, sufficient amount of surfactant form, which is adequate to support the extrauterine life, and by approximately 34 weeks of gestation, surfactant levels generally adequate to support extrauterine life. Finally, the alveolar stage, which ranges from 36 weeks to postnatal life around 8 years, and during this stage, alveoli continue to multiply and mature with progressive improvement in gas exchange efficiency. Now this slide is showing the pulmonary surfactant life cycle. It involves 4 key steps. First is the synthesis and storage, and synthesis occurs in the lamellar bodies, which are the organelles in the type 2 pneumocytes, and these lamellar body then secreted by exocytosis into the alveolar space, and these then convert into the tubular myelin, which is the functional form of the surfactant. Then this tubular myelin forms the monolayer of surfactant to reduce the surface tension of alveolar fluid inside the alveoli, and this prevents the alveolar collapse. Finally, surfactant is either recycled back by the type 2 pneumocytes, or it can also be degraded by the alveolar macrophages to maintain the dynamic equilibrium. Now come to the composition of pulmonary surfactant. Pulmonary surfactant is composed predominantly of lipids, which forms the 90% part of it, especially the phospholipids like DPPC, which is dipalmityl phosphatidylcholine, and this DPPC is the main phospholipid or lipid component of the surfactant, which is primarily responsible for reducing the surface tension. Neutral lipids like cholesterol also found in the uh, pulmonary surfactant. It helps in the packaging and fluidity of the lipid film. And other components are the protein components, which form approximately 10% part of the surfactant. There are two types of the proteins. One is hydrophobic protein and other is hydrophilic protein. Hydrophobic proteins helps in the adsorption and stability of the surfactant to maintain the film over the alveolar surface, and hydrophilic proteins are helps in the defense and regulation of the innate immunity in the alveolar fluid. The functional importance of surfactant is best understood through the law of Laplace, which states that the collapsing pressure P is equals to the or directly proportional to the surface tension and inversely proportional to the alveolar radius r. The collapsing pressure is actually the inward pressure which act over the alveoli from outside to inside to collapse the alveoli. And in normal lungs, the surfactant reduces the surface tension, um, especially in the smaller alveoli, to equalize the pressure and preventing the collapse of smaller alveoli. Because in the smaller alveoli, r is small, so the P is high. The inward pressure which is acting on the smaller alveoli is higher than the collapsing pressure P on the larger size alveoli because of larger radius. Surfactant prevents the collapse of the alveoli, especially in the smaller alveoli. In cases of neonatal respiratory distress syndrome, where surfactant deficiency leads to uniformly high surface tension, causing smaller alveoli to collapse, leads to atelectasis of these smaller alveoli into the larger ones. So it results in the impaired gas exchange and respiratory failure in cases of neonatal respiratory distress syndrome. Now come to the fetal lung maturity tests. Fetal lung maturity tests are used to assess whether the fetus has adequate surfactant production for effective respiration after birth or not. And first test is the lecithin to sphingomyelin ratio. If this ratio is more or equal to 2 to 2.5, then it indicates the maturation of lungs, as lecithin increases with gestational age, while the sphingomyelin remains relatively constant. Next is the presence of phosphatidylglycerol in the amniotic fluid. Phosphatidylglycerol is a specific surfactant component that appears especially in the later gestational period, and it is suggestive of lung maturity, and it is a very specific test. So it is a very good test for detection of lung maturity. And next test is the lamellar body count, which directly measures the surfactant containing particles in the amniotic fluid. If this count is more than 50,000 per microliter, it is suggestive of adequately matured lung. Fourth is a bedside test, which is known as foam stability or shake test. Is a functional assessment in which stable foam formation after shaking with ethanol indicates sufficient amount of surfactant has formed till now, and in the absence of foam, it suggests the immaturity of the lungs. So collectively, these tests guide us for the maturity of the lung and the it predicts the respiratory readiness of the fetus. Now come to the review questions. Question 1. A 29-week gestation male infant is born via emergency cesarean delivery due to placental abruption. Shortly after birth, the neonate develops significant expiratory grunting, nasal flaring, and intercostal retractions. An arterial blood gas shows respiratory acidosis and hypoxemia. The physician explains that the condition is caused by a deficiency in substance that normally reduces surface tension within the alveoli. Which of the following best describes the primary composition of substance mentioned by the physician? Uh, as we have discussed in our previous slides, surfactant is 90% lipid and 10% protein. Lipid, especially the phospholipids, form about 80 to 85% of the surfactant, out of which DPPC, that is dipalmitoyl phosphatidyl choline, is the major determinant of the surfactant activity to reduce surface tension in the alveoli. So, answer should be phosphatidylcholine B. Surfactant protein A, as we have seen in our slide, is serve as an defensive function, defensive function along with this sp protein D. So spa and D are protective and spb and C are help in spread of surfactant. Another component is phosphatidylglycerol, which is also seen in the surfactant, but phosphatidylcholine is the most important major factor in the surfactant to reduce surface tension. Cholesterol improves the fluidity. Next question number 2. A 32-year-old woman at 31 weeks of gestation presents to the labor and delivery unit with the regular uterine contractions and cervical dilation. To accelerate fetal lung maturation, the physician administers a course of intramuscular beta-methasone. This medication works by inducing the maturation of specific pulmonary cells and increasing the production of surfactant. Now question is, which of the following cell types is primary site of surfactant synthesis and storage? This is an easy question. Type 1 pneumocytes are the most abundant pneumocytes in the alveoli. These help in the gas exchange. Alveolar macrophages, these act as defense system and engulf the bacteria, dust, pollutants to protect the lung alveoli. Goblet cells, these cells produce mucus and the protect the lungs from larger particles especially. And last is the Clara cells. Clara cells are also known as club cells, and these are non-ciliated dome-shaped secretory epithelial cells located in the respiratory bronchioles and terminal bronchioles. So these are not found in the alveoli, these are located in the terminal and respiratory bronchioles. And they have a protective function in the bronchioles, and because they secrete a protein which is known as CC16, which is a protective protein uh, and which has an anti-inflammatory and immunomodulatory function. These Clara cells are also acts as a stem cell in the uh, bronchial to regenerate the bronchial epithelium, and these cells also helps in the detoxification because they are abundant in cytochrome P450 to protect the lungs. And so our answer is type 2 pneumocytes. These are cuboidal cells with microvilli. In these cells, the surfactant produced in the lamellar bodies and secreted by exocytosis in the alveolar fluid. So this is all for today. Thank you.